Yeah, that was
great.
really good.
All right. Well, thanks you guys again for the opportunity. These are topics I could talk about all day, but luckily you're just going to be limited to 20 minutes. Elk hoof disease is a disease that probably many of you are familiar with. And is present, of course, here in the Northwestern United States. Chronic wasting disease is one that if you're in Washington, it's knocking on our door. And so it's really important to be aware of these diseases. So I'll just give you a super quick introduction and try to focus on things maybe you should think about with surveillance. So elk hoof disease, like I said, many of you are probably aware of it. The characteristics of the disease are these lesions on the feet that can become severe. We can see overgrown or misshapen or even sloughed hoof capsules. And those are associated usually with ulcers and kind of rotting or necrosis on the sole of the foot. And associated with that, these elk often become debilitated, can have a higher likelihood of mortality. And uncommonly, it's uncommon that we would see recovery from this disease when we see lesions as severe as these. Fortunately, the disease has only been identified in elk and not in other wildlife species. One thing I really wanted to point out was the range of lesions, though, that can occur. When we think about elk hoof disease, people usually think about those really ugly lesions when you get sloughed hoof capsules, like's on the right of the screen there. But it's really important to also be looking for less severe lesions. And so looking between the toes on the animal. So pull the hooves apart, clean the mud or whatever from between the toes and have a good look, because that's where the lesions start. And they can be rather minor initially, but we don't want to miss those when we're doing surveillance. The disease is often thought of as one of occurring in southwestern Washington, and that is where it originated, but it has a much broader impact and range than just southwestern Washington. In 2014, I believe it was detected in Oregon. It was about 2008 that it was detected in southwestern Washington. And it was pretty much thought to be fairly local. In 2018 to 2020, though, we did some additional enhanced surveillance working with states and tribes and federal agencies and saw that the disease has a much broader distribution than we originally realized. And we found that it was also found in Idaho and Northern California. These red plus signs indicate where locations where TAD has been, or elk hoof disease has been found, but it's not showing every case. And I will point out that we do have some areas where we're pretty sure it occurs, but we haven't received hooves from those areas to actually confirm the disease. And so there might be a few gaps. And if you see a gap in it, for example, an area where you live, that's an area where we could really use some help with surveillance and getting more samples in. So a little bit about the disease itself. The lesions are associated with a type of bacteria that's a spiral-shaped bacteria called treponema. And these treponema species are similar to those that are found in a disease of cattle called bovine digital dermatitis or hairy heel wart. And at this point, we're not sure if it's the same disease in both of these animals or if they're separate. But what we do know is that some of the bacteria that we see are the same in elk hoof disease as what we see in bovine digital dermatitis. Because of the presence of these treponema species, the disease has been called treponeme-associated hoof disease, or TAD. The way the disease is diagnosed, I just wanted to point out, because that slide in the lower part of the slide there is what a pathologist looks at. So that's a section of tissue that has been prepared with a silver stain. And then the pathologist has to scan that slide and find little spirally shaped bacteria to diagnose the disease. So it's quite labor intensive. While we diagnose the disease based on the presence of these treponema, there's also information that suggests this is probably a disease that is caused by multiple bacteria in addition to treponema, but we don't know at this point. And so we need to do more investigation on the cause of the disease. Stakeholders are usually really concerned too, in addition to these bacteria, about nutritional deficiencies or mineral deficiencies, or in some cases, herbicides and fertilizers from forestry practices that they believe are strongly contributing to or causing this disease. And so we need to keep those things in mind. And I'll show you a couple of experiments that we're doing that hopefully are starting to get at that. At WSU, we were in 2017 given the lead by the state legislature to lead research into this disease because it's so much of a concern. And so we have 4 areas that we're looking into: the disease distribution and risk factors. I've shown you the map already that we have produced. Looking at the disease agents and new diagnostic tests so we can have more rapid diagnostics. And then a real cornerstone of our work is we built a research facility and we're doing work with captive elk under controlled situations where we can learn more about the disease. And then also the importance of the social aspects of the disease, keeping stakeholders involved, and also really listening to the questions that they have. And today I'm going to be talking about 3 of our research projects that were really spurred by stakeholder interest. The first one was transmission of the disease. Is it actually an infectious transmissible disease, in which case we will manage the disease one way, or is it caused by toxins, say from the forestry practices? And so what we did is housed clinically normal elk in our captive facility, and we made up an inoculum, that picture on the gauze up there in the top left-hand corner, of hooves from affected animals that were chopped up and mixed with soil. And then we put some of that on the floor of the pen where the elk lived. And then we took some more and we put it between their toes. And we put this light little wrap in place to keep the inoculum in place. And what we found is that we had 4 treatment animals and then 2 control animals. The 2 control animals were treated identically, except the inoculum that we made was made from clinically normal elk hooves that had been autoclaved to kill the bacteria in it. And what we found is that 4 out of a 4 treatment elk, but neither of our control elk, developed gross and histologic lesions that were indistinguishable from those that we see in free-ranging elk with TAD. And we also were able to use DNA methods to identify treponema and some other bacteria that are commonly associated with TAD in those lesions. The lesions occurred and were persistent by about 4 months after we started challenging the elk. By 5.5 months, these lesions had become quite severe. Elk were starting to lose body condition, not be as active, and 3 out of the 4 of them reached humane endpoints and were euthanized. Surprisingly, though, 1 recovered. But I think it's important to remember that life in captivity is quite a bit easier than it is in the wild. So we shouldn't rely on recovery in the wild from this disease. So our conclusion was that TAD is transmissible to elk when they're exposed to infective material in soil. So we know that they can be infected from walking on soil. There may also be animal-to-animal transmission as well. And again, this is important because the way a disease is managed is going to be different if it's transmissible versus if it was a toxin. But that said, we know that elk that are otherwise normal can get the disease, but there could be some changes in their susceptibility. And one of those things that could change susceptibility is their mineral status or their nutritional status. And so we opportunistically looked at their mineral levels of elk that we had been following in our surveillance work. And all we get is usually, some of you guys have submitted those feet, a foot that's cut off at about the level of the fetlock. And so those are really all the samples we have to work with. So that's really great for diagnosing TAD, but it's hard to do much more with. Generally, if you're going to look at mineral status in an animal, you like to look at an organ like a liver. But what we do have with those feet that were submitted for diagnostics is hair. So we clipped the hair and ran mineral analyses on that. And then we did multiple regression models to look at some of the different minerals to tell us if any of them were associated with the occurrence of TAD or were protective, if you will. For example, selenium. That was the one that came out important, and this is a really busy slide, but what we're doing is looking at those minerals, those variables that had a odds ratio of less than 1, so if over on the left-hand side of this graph. And selenium was the one that consistently came out that if selenium was low, the animal had a higher likelihood of having TAD. Now, that's something that's of interest for pursuing farther, but at this point, I'm not ready to say, like, go out and start supplementing elk with selenium to treat this disease or prevent this disease. And we need to do more studies. We're doing one looking at liver levels. We've gotten a batch of livers that we'll be able to look at and have additional information. But the problem with doing something like supplementing is that we could be congregating elk and actually promoting transmission of the disease while we're hopefully trying to treat them. So stand by on that one. Another question of stakeholder interest was the occurrence of asymmetrical antlers that people were seeing in elk with hoof disease. And so we were able just to use some data that already existed that Washington Department of Fish and Wildlife collects from hunters on antler point counts and the game management unit where they harvested their elk, and then a question that asked if there is the presence or absence of hoof abnormalities. And what we found was that, sure enough, stakeholders were right, that elk with elk hoof disease, or TAD, had double the odds of having asymmetric antlers as ones without TAD. And so it's another indication that how important stakeholder observations are and the additional impacts that this disease can have, because antlers are important to people and to elk. So a few take-home messages to kind of sum up. We've got now good evidence that TAD is a transmissible infectious disease and it's associated with Treponema species. Therefore, management actions that address reducing transmission are appropriate. We still have more work to do, though, on the cause of this disease. The lesions are generally limited to the feet, but we know that this now antler asymmetry can occur. In general, we don't see in the elk that we've looked at impacts on the meat, but that can really vary with how debilitated and run down an animal is. So caution should always be used there. We know that this disease is not just a local issue, it's a regional issue, and it's one that could continue to spread farther. And so surveillance is really warranted, particularly in those areas where we haven't diagnosed in the laboratory to confirm TAD in those areas. So if you're doing surveillance, particularly in those areas, be sure and look at the feet of animals that you see. And remember, don't just look for those terrible lesions, but look between the toes and look for those mild lesions and submit those for laboratory confirmation in new areas. And don't expect anything as far as a treatment, an antibiotic or a vaccine or anything. The treatment, if you will, is going to be through the way we manage these animals. So now going from a disease that we do have in Washington and the Northwest to one that is knocking at our door and is really a concern. Chronic wasting disease is a disease of the deer family, and it is characterized by emaciation or animals that get really, really skinny, which as you can see in this elk. They have abnormal behavior and listlessness, and I call it kind of a 100-mile stare. There's just nobody home when you look in their eyes. They can also have excessive salivation as another sign. CWD, as I mentioned, has not been detected in Washington yet. About a little over a year ago, it was detected in Idaho, so it is really right up against the border of Oregon and Washington. And when I started my career studying chronic wasting disease in the late 1990s, chronic wasting disease occurred in 2 states, Colorado and Wyoming. And now I think we're up to about 30 states in the US, and 3 Canadian provinces have detected CWD in free-ranging deer and elk. So it is a bad disease. It is a fatal neurologic disease. Recovery is not known to occur. It's in the family of diseases called the transmissible spongiform encephalopathies, or TSEs. Other diseases in that family include scrapie in sheep and bovine spongiform encephalopathy, or mad cow disease. And these are diseases that are not caused by a bacteria or a virus or a parasite or a fungus. It's caused by an abnormal conformation of a prion protein. This prion protein is normal in your body, in the body of animals, but when they change their conformation, then they accumulate and they're not broken down. And what we see is more or less holes in the brain. And the disease only naturally affects members of the deer family. So in North America, elk, mule deer, white-tailed deer, and moose have been diagnosed. In some other countries, reindeer, or if you will, caribou, have had the disease as well.
Margaret, you have 3 minutes left.
Okay, thank you. It is a prolonged course of disease, so it takes the animal a year and a half before they'll really start showing these end stage signs like we see in these pictures. In deer, males are more susceptible to disease, but in elk, there doesn't seem to be a sex predilection. Again, there's no vaccine, no treatment, no way to control chronic wasting disease except through management. The disease is spread primarily probably through carcasses or excretions from live animals, saliva, feces, and urine. These get into the soil and stay for a long time. Prions don't break down, and so this environment is contaminated with these prions for a long time, and disease transmission can occur. To diagnose the disease, we generally need to look at the brain or the lymph nodes in the head of the animal. And those need to be submitted to a diagnostic lab. And there's a rapid, if you will, ELISA, that only takes a day or 2 to do. It's not like an animal side test. Or the animal, the tissues are looked at under the microscope using a special stain to see the accumulation of prions. We really can't reliably diagnose the disease in live animals. We need the head of the animal after death to make the diagnosis for sure.
2 minutes.
Okay, so again, some final take-home messages. I would argue this is the most important disease in wildlife. It's a unique class of disease, being a prion disease, and these environments are contaminated for a long time once they become contaminated. We really need to be doing surveillance, particularly in areas where the disease hasn't been identified yet. If you ever have an animal that you think might have chronic wasting disease, the best thing is to take the head. There's a portion of the brain and retropharyngeal lymph node, it's called, that can be submitted for testing. Remember, don't shoot the animal in the head if it's a CWD suspect, because we want that brain tissue intact. The disease is not known to be transmitted to humans, but CDC recommends that these animals, if they're CWD suspects, not be consumed by humans. And if you'd like to know more, there's lots and lots of literature out there on CWD now, but Escobar has a nice review paper. So if you have additional questions about surveillance or about submitting samples, please let me know, and I'm always happy to talk more.